Today we're going to be talking about macromolecules. It's a biology concept that is really focusing on the molecular side of biology and how things are actually moving around and what cells contain. Macro means big. The term macro means big. If you're talking about macroeconomics or things on a macro scale, you're talking about things that are happening on a big scale. So macromolecules literally means big molecules. And macromolecules are really the building blocks of cells. Almost everything inside of a cell is made up of some sort of macromolecule. There are 4 classes of macromolecules. The first one is a carbohydrate. Carbohydrate. The second one is a lipid. Then a protein. And lastly, it's a nucleic acid. These are the 4 major classes. These are the most common macromolecules. Most common. The first one that we're going to talk about is a carbohydrate. So a carbohydrate has a main function, and that is it is the energy source. It is the energy source of a cell. So if we have a runner such as Usain Bolt, and he is running along his track. So if there's a track here, and someone like Usain Bolt is running along the track, a macromolecule, that a carbohydrate is the energy source. So that is actually how Usain Bolt is running. So a carbohydrate, basically you get energy from it by using it in cellular respiration. Cellular respiration. And how do we use carbohydrates? Well, some examples of carbohydrates are glucose. Glucose is a carbohydrate. Sucrose. And starch is actually a carbohydrate. So glucose is what we see when we do cellular respiration, and we say that, oh, well, I need to take a molecule of glucose, and then add some oxygen to it, and then some water, and I get ATP. So that's what glucose is used for in cellular respiration. Sucrose doesn't sound very familiar, but it actually is. It's actually table sugar. So the kind that you would put on inside of a coffee or in a cake is sucrose. And starch is the kinds of things that we find in potatoes. Usually, potatoes is an example. Starch is usually found in plants. They're actually long-term energy storage they're actually the energy storage of plants. So actually, each of these 3 things represent a type of carbohydrate. This is called a monosaccharide. And a monosaccharide is actually a simple carbohydrate, a simple sugar. So those are these are the quick burn kind of things, and you know that because glucose is the very base molecule in cellular respiration. So as soon as you got glucose, cellular respiration can start. You don't have to transform it. On the opposite end of the scale, we have starch. Starch is a polysaccharide. And a polysaccharide is the complex form of sugar. It has to be broken up into and basically transformed into a monosaccharide in order to be used. Sucrose is kind of in the middle. It's an example of something called a disaccharide. Disaccharide is sort of made up of 2 parts. So glucose is made up of 1 monomer, and starch is made up of many monomers. And sucrose is actually made up of two monomers. So glucose is the base, is like monosaccharides are the base, and then polysaccharides are the polymers. So like they're the main unit. Now, starch, carbohydrates actually look like this in a when they're all bonded. They look a little bit like this in a hexagonal form. But what we see here is an example, for example, is glucose. Glucose is C6H12O6. Now, all carbohydrates have this C, H, and O. And all carbohydrates have C, H, and O in a 1 to 2 to 1 ratio. That is one of the key identifiers of a carbohydrate. You can count up the number of carbons, hydrogens, and oxygens. And if that is in a 1 to 2 to 1 ratio, then you know for sure that it's a carbohydrate. So when you have it in a hexagonal form like this, you can see that this is how all of these carbons, hydrogens, and oxygens would arrange themselves. Usually the carbons would arrange themselves on these vertices right here. This would be the carbons that I'm drawing in, and then the hydroxide, and there'd usually be hydroxides somewhere over attached to these. And then there are also hydrogens that line the insides. So that's how you get the H12. The next type of monomer is the next type of macromolecule is called a lipid. A lipid is, we know most lipids because they're greasy. They usually come off of a food takeout bag. They're fats and oils. We also know them because they are steroids. And they are also phospholipids. They're also phospholipids, and phospholipids we know because of the cell membrane. They're what make up the cell membrane, so that's how we know phospholipids. And these are the different polymers of lipids. And there are two monomers. So these are the three types of polymers. And the monomers are actually two things. We have the fatty acids, and then we have the glycerols or glycerols. These are the monomers. So what else? What can we do with fatty acids and glycerols? Well, I'll show you how we make fats and oils. A fat is called a triglyceride. It's pronounced like this, triglyceride. Spelt like this. And it actually looks like this. And if you were to look at it under a microscope, you would see something probably like this. But if you look at the chemical formula, you'll see that we have the glycerides are actually long chains of either saturated or unsaturated fats. Remember that the definition of a saturated fat has the most amount of hydrogen bonds possible, so no carbon-carbon bonds. So we put the hydrogens on either side. This is just the structure. We have an oxygen, and then we will have an OH right here. And then that's the structure of fatty acid, but when you actually bond it, it looks like this. And then we have, right here, actually, we have carbon, hydrogen, And then oxygen is what bonds these two together. And then this actually repeats 3 times. So if we take what I just did here and we multiply it by 3, so we basically cut and paste these things 3 times, we get what a fatty acid would look like, what a fat and oil would look like. Now, a steroid actually is a whole bunch of rings. That's how you identify what a steroid is. You have many, many rings. Actually, you have 4 rings that are formed. That would be a steroid. And a phospholipid is actually this right here, the fatty acids. And when you do a phospholipid, you actually remove one of the fatty acids and attach on a phosphate group. Now, this phosphate group actually makes the fatty acids slightly polar. So that's how we make the cell membrane. The heads of a phospholipid are polar. So if I'm going to draw a cell membrane, these are the heads. These are polar / hydrophilic, which means they love water. And these are nonpolar or hydrophobic. In a later video, we're going to talk about the other two, proteins and nucleic acids. And then we'll also talk about how we're going to test for these things. There are certain tests that you can do, such as a Bayer's test or a Benedict's test. So we'll learn about that in a future video. Thanks for watching.